What is DNA replication? As a definition, it is a process by which DNA makes a copy of itself during cell division. That means, as a result of DNA replication, we will get two identical DNA molecules. Why do DNA replicates? Every living organisms need to grow. For that, there must be cell division. At the time of cell division, every cell content should be equally divided, including DNA. There comes the role of DNA replication. In other words, DNA replicates in order to transfer same genetic information into two new daughter cells. Let's see process of replication. Several enzymes plays in this process. The first enzyme you need to remember is helicase. Helicase is an enzyme that unwinds the DNA molecule and breaks the hydrogen bond between nitrogenous bases and creating Y-shaped region called replication fork. And each of the newly formed strand act as a template to form another strand. Now there is single strands of DNA with single base, and these base pairs of each of the strands are complementary to each other, so there is a chance to bind it again. But we need to prevent this binding, otherwise no new strands will be formed. For that, SSB will act, which prevents binding it again. SSB is single stranded binding protein. But another strand should also be formed with complementary base pairing. Base pairs are adenine, guanine, thymine, and cytosine, in which adenine always be paired with thymine with double bond and guanine always be paired with cytosine with triple bond. In order to add nitrogenous bases, DNA polymerase enzyme is needed, but DNA polymerase enzyme doesn't start adding nitrogenous bases by itself, but it needs a primer, and the primer is released by primase enzyme. And an important point to be noted is the new strands are always formed in 5-3' direction. DNA polymerase 3 binds with the primer and starts adding complementary nitrogenous bases. As the DNA polymerase moves, the single-stranded binding protein gets disappeared and the new strands are formed again and again with single primer. Since it is a continuous process, it is called leading strand. On the other hand, in the opposite strand ends at 5' end, so the adding strand must be 3'. But nitrogenous bases are not going to add in 3', 5' direction, so the primer is released somewhere here and form the strand. Again, another primer is released somewhere and continue the process. This is called lagging strand, and in this strand the fragments are called Okazaki fragments. On the other hand, lagging strands needed more and more primer released somewhere and continuing the process. In the leading strand, there is only one primer and it will be removed by RNase. But in case of lagging strand, there is more than one primer and this also will be removed by RNase. In lagging strand, there is discontinuous fragments. It needs to be joined. So this will be facilitated by DNA ligase. Let's conclude. DNA replication is a semi-conservative process. That means only one strand of the parent DNA is conserving for the daughter DNAs. Helicase enzyme unwinds the DNA and breaks the hydrogen bonds between nitrogen bases. SSB, single-stranded binding protein, prevents the nitrogen bases from binding again. DNA polymerase 3 helps to add nitrogen bases to form the new strand. Primase enzyme helps to release primers as an initiator for the process. RNase H helps to remove primase from both leading and lagging strands. DNA ligase enzyme joins the discontinuous fragments of lagging strand. And now we got two daughter DNAs as a result of replication. Thanks for watching. Please do like, share and subscribe.